麦克风就这个是吧 ？Okay. So it's my great pleasure to be here to give you a talk about virtual cell. So I'm Qi Liu, Liu Qi, from Tongji University in Shanghai, in China. So before my talk, I would like to give you a brief introduction of my research group. So my group is just focused on AI for precision medicine study. So we call it AI for PM. So basically, we just using two technologies to address very exciting clinical problems. The first one is AI, and the second one is omics technologies. So we think when we first focus on omics data analysis, we will encounter a lot of challenging issues. So we have to develop a lot of AI technologies to address the issues here. Also, we have a very great research platforms. So in Tongji University and in Shanghai. So we have the bioinformatics undergraduate programs in Tongji University. And we also have the joint research center between Shanghai and Tongji University. Okay. So we also write a book about omics machine learning. In Chinese, is 组学机器学习. So in this book, we just summarize the cutting-edge machine learning technologies for omics data analysis, and also we demonstrate a bunch of clinical applications to using AI and omics technologies. Okay, so today I will talk about AIVC. It's called AI virtual cell. So I think this topic is recently a very hot topic in the community. And recently, Cell just published a leading edge perspective article. It's talk about how to build a virtual cell with artificial intelligence, the priority, and the opportunity. So this article just summarizes I think it's very brief definition of what is virtual cell and what is AIVC. So for virtual cell, I think is basically is a kind of in silico modeling to simulate, to predict, and to steer the cell behavior. And regarding AIVC, in my understanding, is more focused on the application of AI and omics technologies to the cell modeling. It's kind of construction of the cell models, just learning from the sketch. It's just learning directly from the omics data. So basically, when we talk about AIVC, it's a kind of to apply the multiple scale, multiple modality, and often the large deep neural network, for example, the transformer-based model, to represent and simulate the behavior of molecular cells or even the tissues across diverse states. So this is a kind of two different definitions for virtual cell and for AIVC. Okay, so it also summarizes the utility of AIVC. So from my understanding, it's kind of lab in a loop schema based on AIVC. So for example, you can couple the in silico modeling with the experimental validation and feedback to form a loop, and using such kind of loop schema to apply it in drug discovery to disease modeling. Okay, so I just summarized the three key capacity of the AIVC. The first one is multi-modality measurements. And the second one is multi-scale representation. So a virtual cell should be represent from multiple scale, from the molecular level, from the cellular level, and from the multiple cellular interaction level. And the third one, I think is the most important one, is talk about the dynamic modeling. So a virtual cell should be capable of modeling the dynamic response of the cell on the different perturbations. So here is I think is the 3 key points of AIVC. Also, this perspective articles also present, I think is a very fundamental issues about AIVC. That is the representation of the cells. So this article just present a notation. It's called UR. That is universal representation. So here is a comparison between a traditional physical cell and AI virtual cell representation. So you can see here the right part. So the AIVC representation just involve the representation of the molecular level, the cellular level, and the multicellular interaction level. So for example, we can using the graph structure. For example, the GNN, the graph neural network modeling the different interaction between different cells. So here, we just raised the key point of my talk today. So I think this is also the fundamental issues to study AIVC. That is, we should perform representation learning for AIVC. But the question is how? So first, let us to see the mainstream representation learning models exist in the community. I think the most mainstream representation learning model is foundation model, right? And the foundation model is very powerful in its representation ability, right? And we have witnessed a bunch of foundation models in this community. For example, the Geneformer, the SCGBT, the SA Foundation, blah, blah, blah. We have a bunch of foundation models. But I think all this foundation model is actually a black box representation. In Chinese, it's 黑盒表征, right? So although it's very powerful in its representation, but it has several limitations. So I just list the limitations here. The 4 limitations here. Number 1, such kind of representation is entanglement of biological signals. Number 2, such foundation model-based representation just commonly lack of multimodality integration ability. Number 3, the foundation model often handle the snapshot modeling without dynamic model capacity. Number 4, foundation model is obviously short of causality interpretation ability, right? So I just list a bunch of limitations of the representation from foundation models. So that's why we want to propose a kind of, we call it white box representation of AIVC. So besides foundation model, we want to perform white box representation. So in Chinese, 白盒. But how? How to perform white box representation, right? So in our study, we just performed some preliminary study, and we have developed a computational framework. We call it causal cell. And we want to propose a very, I think it's very efficient and a very, I think it's very inspiring representation methods. We call it disentanglement learning. So, okay, so this is my talk. the key point of my talk today. So we also summarize for functionality a white box AIVC should have. The first one is interaction perception. The second one is modality integration. The third one is perturbation prediction. And the final one is disentanglement generation. So today I will just share the last one with you. And I also would like to thank Dr. Chen Qian here to handle our manuscript and give us this great opportunity to stand here to share with you my, I think it's a very interesting story. Okay, so basically we just developed a computational framework called causal cell. And we want to perform causal disentanglement for single cell representation and controllable counterfactual generation. Okay. So the basic idea of this computational framework is we want to perform the causal disentanglement. That is just to decompose the entanglement biological concepts behind the phenotype of the data, right? This is just like you cut the Gaudian note. The Gaudian note is just a saying from Greek mythology, right? You want to cut the note, right? So we have developed the framework called causal cell. So basically, causal cell contain two parts. The first one is we want to perform causal disentanglement. So basically, we want to transfer the phenotype omics data into a concept-based representation of the cell. And during the transformation of the phenotype data, we will incorporate a kind of prior constraint. It's called structural causal modeling. So that means we want to incorporate certain causal structure constraints to guide the transformation procedure. And on the other hand, we can also perform counterfactual generation. For example, using the generative AI model. For example, in our study, we have using the diffusion model. And such kind of generation is a kind of de novo generation of the new cell samples. But this kind of generation is not arbitrary. It's also guided by the structure, by the causal structure, also guided by the prior to make sure we generate the biological meaningful cell samples. So it's not a kind of arbitrary generation, right? So just to make sure we generate the cell samples just to follow the biological constraint, right? So this is the two parts of the causal cell. Okay, so here is another illustration of our model, right? So basically you can perform this entanglement representation of the cell, and you can get a bunch of concepts embedding, right? And also you can perform counterfactual generation. Basically you can manipulate the concepts, and you can generate new cell samples by combine different concepts together. That means you can generate new cell samples, but it's biological meaningful, right? And here is we just using a single cell data set to demonstrate our disentanglement performance. So this is a single cell data set, just the single cell just under treatment by ICB, it's immune checkpoint blocker. So basically, you can see here, among this data set, you have a bunch of biological concepts. So our study just want to decompose this biological concept, and we want to benchmark the disentanglement performance in different experimental settings. We just benchmark it in two different experimental settings. One is we call the in-distribution splitting. That is, the model has seen all possible combinations of concept labels during the training. We just tested it in a similar or identical distribution. And the second one is we call the out of distribution splitting. So that means the model will encounter unseen combinations of concept labels in the testing. So obviously, the second one is much more challenging testing, right? So you can see in both cases, compared to the baseline models, causal cell has achieved the best disentanglement performance. Okay, so we're also using the concept embedding to perform the clustering of the cells to annotate the cell types. So in both cases, in the fine grain and in the coarse grain setting, you can see our model is also achieve the best clustering results compared to the baseline model. And also, we benchmark the reconstruction performance of our model. That means we want to generate biological meaningful new samples. And we compare our model with traditional disentanglement model and the generative model. For example, the SC gene, SCVI is also very mainstream single cell generative model. And we benchmark all these existing tools in a bunch of evaluation metrics. You can see among all the tools, we also achieve the best reconstruction performance. Okay, here is another I think is a very interesting case to demonstrate the causal structure consistency in the counterfactual generation. So we just focus on data set. This data set is actually a spatial temporary live data set, just investigating the host and the parasite temporal expression program. We just inject the parasite into the host cell to see whether the host cell has infected or not. And we have the ground truth, right? We have defined a score to indicate the infected status of the host cells. And we have the ground truth, that is only the injected and the infected cells should present the highest score. And we test two different cases. The first case is without causal prior, without causal constraint. We just perform the causal disentanglement and counterfactual generation. And we generate the data. And you can see in this case, we just generate some of the cells that is not make sense, right? Because we find, you can see here, actually the cell is infected. The cell is injected but not infected. So this case should not have such high score, right? So this means that if we didn't consider the causal prior, we have not, obtained several cases that is not make sense. But if we consider the causal prior, we actually get the results that is just match the ground truth, right? Only the injected and the infected cells should present the highest score. Here is another very interesting data set to demonstrate that the counterfactual generation can be used for find novel biological discovery in a very, very small sample case. So in this case, we just look for the mouse aging data set. This mouse aging data set is actually a morphish brain data set, including a very small number of mouse samples during aging, during 3 points in the, you can see here in the 4 weeks, in the 24 weeks, and the 19 weeks. 3 different time points. And all these genes, all the 3 genes is actually the ground truth gene markers that is related to the neuroaging. And you can see here, if we're using the original small samples, it cannot capture the real gene expression change, and it cannot identify the ground truth gene markers. But if we're using the counterfactor generate small virtual samples, it do capture the real gene expression change and identify the ground truth gene markers. Also, we're using causal cell to identify a very novel gene that has some biological functions. So this is, I think, is very interesting case just to show you that we can using such counterfactor generation schema to find a novel biological discovery in a very, very small sample cases. Okay. So, okay, I would summarize the 3 key contributions of causal cell in AIVC study. The first one is explainability. This is achieved by causal disentanglement. The second one is generalizability. This is achieved by diffusion model-based generation. And the third one is controllability. This is achieved by counterfactor generation by controllable manipulating the biological concepts. Okay, our work is just highlighted by the computational and theoretical biological connection. So thanks again for Dr. Chen Qian for giving me the opportunity to share our work. And also, our work is just comments by social media. So I just like these comments. So it says, imagine that you have the ability to play what if with cells. So it sounds very cool. So I just like these comments. So, okay, I will end here. But I think it's not the end, but the start. So let's make explainable, generalizable, and controllable AIVC. So I would like to end by the famous saying by Richard Feynman, what I cannot create, I do not understand. Thank you, that's all. Thank you.
Thank you. Question time.
Well, thanks a lot for the impressive talk and the latest work. So far, we're talking about data-driven AI a lot, like both causal inference or deep learning or what. And in systems biology, I think we also have some curated knowledge or open access knowledge bases built by previous domain experts, like ontologies or genome scale metabolic models or gene regulatory models. And I think they are also quite vital to build a, you mentioned human comprehensible beyond just simply simulation or prediction. And so what do you think this kind of existing knowledge-driven methods can contribute or can integrate with our existing data-driven learning, AIVC, or cell modeling?
Sorry, I cannot hear clear what is your question here. So can you just repeat the last sentence? So what's the question here?
Sorry for that. Can you hear it clearly now?
Yeah, a little bit, yeah.
What do you think we are gonna integrate or use existing knowledge to contribute our cell modeling or current data-driven virtual cells?
So your question is, what do you think to incorporate the prior knowledge into the building of cell? This is your question? Right, I think it's important, right, very important. So the key point here is just your question here, right? Okay, thank you, yeah.
we have a lot of like gene ontologies.
Yeah. Gene ontologies. Also some prior relationship interaction. All these things can be incorporated as a prior knowledge to incorporate in your study, in your modeling, right? Okay.
Okay, thanks.
Thank you. Yeah.
Yeah, wonderful work.
Oh, Professor Huang. Yeah.
Excited for your very inspiring. So I'm wondering, do you have any like a future plan to align all the key elements that you mentioned, like a perturbation, dynamics, or something? Yeah. Thank you.
Yeah. So actually, I think today my talk is just focus on representation and not too much touch the dynamic modeling. But I think dynamic modeling is very important for virtual cell construction. And so basically, I think a good representation is definitely will boost the perturbation prediction, right? So recently, maybe if you're familiar with this area, you will see a bunch of papers talk about if you're using foundation model-based representation for gene perturbation prediction, actually the performance is not just good as even not good as a linear regression, right? So that means actually representation is very important. Yeah. Okay, thank you.
Thank you very much. It's very inspiring. So I have a question about these white books and also for the understandability or interpretation ability. So I think maybe everyone knows to understand is very important, but it still depends on to which extent of the understandable. For example, if we think deeply about the physical equations, for example, the Einstein's equation, the energy is equal to the mass multiplied by the speed of the light speed, but we cannot still understand why this energy is equal to this. So I would like to ask is to which extent do you think is acceptable level of the interpretability for this AI foundation models, for this biological AI foundation models, for example, the cell representation, at the gene level or signaling pathway level or protein-protein interaction level, or to which extent of the level is extendable? Yeah, that's my question. Thank you.
So from my opinion, I think all these things, if you can incorporate, you just incorporate. I think all these things is better for the representation. But, so you just mentioned the physical energy, right? So I think the key difference between biological and biology and the physical chemistry is that in biology, we just short of a kind of theory, a quantitative theory, just like the Newton theory in physical, right? So that is, I think, is a key challenging issues here. So we cannot incorporate too much quantitative rules in the study. But I think that's we sit here, right? So maybe in the future, we should find some quantitative rules from all perspectives to incorporate into our study, right? Okay.
Okay, so can I just reply to this, or we can discuss it during the coffee break?
Okay, thank you.
Because of time limit, and you can reach out to the speaker during the coffee break.
Maybe I have a very small question. Is that, Prof. Liu, can you comment on that? Whether the concept-based models will be a new direction for the foundation models that make the model more explainable? This is a new direction. Like in the future, we are not focused on the.
I think it's not a new direction. It's just the alternative direction, right? Okay. You can use it as a complementary to the foundation model. So I think both paths has its advantages and disadvantages. For example, the causal model is very, I think it's very explainable, but it also has disadvantages. The key challenging issues here is you have to discover the causal structure first to incorporate it into your study, right? This is very challenging, right?
Okay. Thank you. Thank you.
Okay.
Thanks, Ji. Let's thank the three speakers again. Okay, now let's have a coffee